Okay, shall we begin? Ready? Okay. Um, so this session is about generating your own 3D brain model outside of TVB, or more generally, it's about visualizing these kinds of data that you saw in the last 2 days. 3 days? 3 days, 2 days? 2 days. um, and how you could visualize them basically outside of TVB. So this is the topic of the session. I think you already saw a few options to visualize it within TVB. So TVB has also this viewing options. Here we look at some options outside of TVB. And we'll go through 3 examples. The first one is like some kind of, if you want to just quickly visualize the data without much ado, without much coding, there are Python toolboxes for 3D visualization. So it's basically, so visualizing a brain model is nothing else than plotting something in 3D basically. So brain models are based on surfaces, triangulations, and we have nodes and so on. And yeah, we'll look through the code in a second. The second one is a video I made about, I don't know, maybe 6 years ago or so. It was like a first attempt to visualize all these different kinds of data that we have inside a single video. It's still quite coarse and not super beautiful. It's based on MRView, which is comes with MRtrix tractography software, which we use for tractography. And it's just an example how you can use external packages to construct videos. So the software doesn't render super beautiful, one has to say. It's so the Mayavi, in my opinion, also doesn't. So the first one with Python also doesn't really produce the most super beautiful visualizations. So there are not like dedicated rendering options. So the nicest visualization you get with a program called SurfEye. So in my opinion, the nicest, because they really focused on rendering brain surfaces in a nice way. And this is quite a complicated thing to do. So the last video, the third one, is quite complicated. Basically the goal of the session is to show you how you hack your stuff together, basically. So all of this is hacking, more or less. So there's no out-of-the-box solution that lets you do these visualizations. You always have to like combine different packages to get a good result most of the time. Okay, before we show these examples, let's just quickly recollect what kind of data we have with TVB. So the most important data types that we have here. Where is it? Here it is. The most important data types that we have here from TVB, I suppose you already saw them a few times. So if you would want to reduce the input data you need for a brain model, then the single most important data set that you need is such a weights matrix. So this is the structural connectivity. The structural connectivity you can extract using diffusion-weighted MRI tractography. And this is the example data set that comes with TVB, basically. So it has 76 regions, and this connectivity information is stored in a file called weights.txt, and it's basically an ASCII file that contains a 76 times 76 matrix. It's really just one value after the other. So here's a connection value of 2, then again a 2, and so on. So this is a like broken down, simplified version of a connectome. Here we have in this example data set only 4 levels, 0, 1, 2, and 3. And yeah, this is basically 76 columns and 76 rows of floating point numbers. Then the next kind of information you typically supply to TVB are tracked lengths. This is the length of these fiber bands, the average length, and these are used to compute the transmission delay a signal needs to go from one area to another brain area. And so these are lengths in millimeters, and it's the same format that you have in the weights text file. It's basically just a 76 times 76 matrix of floating point numbers. Then we have this file info.txt. It just specifies the units of the weights. So weights are typically dimensionless. We don't have a unit for them. These are relative weights which we need to, where we need to, for example, fit the global coupling scaling factor, a parameter of the model, in order to get some interpretable unit out of that. The length unit, however, is defined. It's millimeter, and the areas is millimeter square. So all of these files, I don't know whether someone showed this to you, so all of these files are basically then zipped as a zip, and then there is an upload function in TVB where you can select the zip and import all of this data. And we have this file cortical.txt. This is just a vector of 76 integer numbers, and there's a 1 if the brain region is a cortical brain region, and there's a 0 if it's a subcortical brain region. Then we have centers.txt. This has 4 columns and 76 rows. The first column is a short ID of the region, and the next 3 columns are x, y, and z coordinates of the center point of that region. The next file is average orientations.txt. This is also a 76 times 3 matrix. So we have again coordinates here, or vectors, which give the average orientation. So if you have a surface representation of the brain, of the cortex, you can define many different normals on the surface, and this value here is basically the average over all normals. Then we have areas, last one, areas.txt, and there we have in square millimeter the size, the area of this brain region, basically the surface area. So this is the mandatory input data set for TVB, and some of this information we will use to plot the model in 3D. And then we have a second data set, which is not mandatory input for TVB, but which contains the cortex surface triangulation. Surface triangulation consists, surface triangulation, the purpose of a surface triangulation is representation of the surface in the computer. And in order to do this, you put dots on the surface in equidistant locations, and then you connect these dots, for example, using triangles, or you can also have rectangles or so on. So surfaces are discretized in this manner using dots and their connections, basically. So, and these dots are called vertices. So in this vertices file, so this is really the entire surface of the cortex, and it's defined by the x, y, and z coordinates of the triangulation nodes or vertices. And second most important part of this data set are triangles. So these are the indices of the vertices which form a surface triangle. So for example, this first triangle on our surface would be formed by the point 0. So this one, then we have 1, and somewhere below 13. So this is like one of the many triangles that are used to specify the surface. And we have also vertex normals. So these are, we still don't know whether this surface is concave or convex. So here we have the normal vector basically pointing outward on the surface. And we have a region mapping. Hello. So this is also just a vector which associates each vertex of the surface triangulation with the brane region that is at this position. So our surface triangulation has 16,000 points. So this is like more or less the minimum to get a more or less smooth or sharp surface representation. And we associate each vertex of our vertices file with a region ID. So the first vertex belongs to region 36, also the second, and so on. So there we know which parts of our surfaces belong to which region. So these are, this is the minimum data set to fully describe such a brain network model, basically. And now we'll look at some Python code. It's just a few lines that basically,
these information that you gave, where can we find them just generally?
So with TVB there is an example data set where all of these files are shipped with. But for your own data you have to construct these files yourself.
But the question is that like the data frame, like this one that you mentioned, just like 1 is what and 0 is what? Is there somewhere that all these are written somewhere? Like exactly the things that you explained now.
Yeah.
Just explanation. Are they in the TVB documents or?
So the files I just showed you, they really come with TVB.
I know, but the format of them.
You mean like a description of this format?
Exactly.
I think this is documented in the TVB documentation. So I don't know. If we like Google for TVB documentation and something like weights.txt, then we find here the site, the virtual brain data formats, and there is some documentation of these files. So
thank you. That's
okay.
Thank you.
Okay, so in general, the documentation is a bit scattered of TVB, so it's easy to miss something. So I think there are 2 or 3 different places where there's documentation for TVB, and often one has to browse through these different pages to find the appropriate information. So it's often not easy to find, but most of these things are documented. So the documentation is good.
Thank you.
Okay, now let's look how we can plot this. So these are really just like 100 lines or so. Have you worked with Python before? And okay, so just ask if something is unclear. So if you worked with Python before, I'm sure you know NumPy, most important package for numerical stuff, matrices and so on. And here we use, for plotting, not matplotlib, we use Mayavi. Matplotlib is not really good for plotting 3D information. So I must say, I know this code now since last week or so. Paul has written it. So I didn't plot this in Python before. I did the other 2 videos. So this is also quite new for me, but it's relatively simple, really. What you do is we read the centers information. So this is the centers.txt file where we have these 3D coordinates of the centers. The region names was the first column of this file, and here we read the weights. So we will show the centers as balls and the connections between these balls as tubes, and we will scale these tubes according to the weight as we find it in the structure connectome. And here we read the surface parcellation. So this is the most important part for 3D objects. So up to now we just had dots and sticks that connect these dots. The nicer thing are surfaces and yeah, for 3D plotting basically. And here we read the vertices, the triangles, and this region mapping file which associates each vertex with its brane region. So here we just make sure that the diagonal of the structure connectome is 0, that we don't have any connections between a brane region and itself. So sometimes when tractography software often also outputs self-connections, and we don't have these here. They are modeled differently in TVB. Okay, this is just a quick heat map to visualize the SC. So important here. Here comes the important part. So this figure function is a function of Mayavi. It opens up a new figure with white background. This is the RGB color model. So we have red, green, and blue here. If we have all 3 primary colors, we get a white. And points 3D is basically a function that plots scatter points at x, y, z locations, which we have here in our centers data structure. And we plot them a little bigger and with red color. And then we have a triangular mesh. This is also a Mayavi function, which we give the x, y, and z coordinates of the surface vertices and the triangles. The mesh will be in white, and it will be quite transparent so that we can look into the inside. So this opacity attribute defines the transparency. Then we select a few regions that we want to highlight. So we will draw these regions opaque and the others transparent. Yeah, just some random set of regions. And what we do here is basically we get our region index. So we look basically in this region mapping data structure. So okay. First of all, we have this region defined by name, right? Then in this file centers.txt, we had also these region names as the first column. So here we look in these region names and we look at which position, at which index is this region. So for example, CCA may be region number 70 or so. So now we know CCA is region number 70. And now we look in this region mapping data structure, we look up all vertices that have an ID of 70, and we select all of those. So we want to colorize all surface vertices that belong to region 70 CCA. Yeah, this is what we do here basically. We start with an empty list, and then we populate this list by adding all triangles that belong to our set of vertices that we want to belong to this region. Then we have a long list of vertices and triangles, and these triangles begin, so the count begins at an arbitrary number. So as we saw before, The first triangle would be between the points 0, 1, and 13. But here now we selected some kind of arbitrary subset of our vertices, which means we need to rename or re-index our triangles that they start at 0, basically, and then increase in 1 steps, basically. So this is just the parameters of text that we plot next to the visualized 3D object. And here we also plot some lines, and here we plot basically tubes that connect the brain regions. And we scale these tubes according to the value in the structure connectome. So if the value is 1, we use a radius of 0.2. If it's 2, we use a radius of 0.8, and so on. So this could, of course, in a more fine, discretized structure connectome, you can find a more elegant way to basically adjust the tube size. And this is really just like a first and simple way to, I already started it because it takes a while until it shows. And this is really, it's not the most beautiful way of representing a 3D brain parcellation, but it has all the essential ingredients that we were looking for. So it has the node centers in red, it has connections between these nodes, these tubes. We have a semi-transparent cortex surface, and we have some regions of interest in opaque. So this is if you want to quickly visualize these data outside of TVB without much effort. Even simpler would be, so this is if you already are comfortable in a programming language like Python or MATLAB or so on. There are also many softwares with a graphical user interface which lets you load parts of these data and lets them visualize. But typically there's no one software package which fulfills all the needs that can really visualize all these different kinds of structures. But at the end we'll look at one which does a very good job, a very nice job at visualizing these. This was basically this first way of quickly visualizing a brain network model.
Sorry, this script is not available, right?
We can package it with the material that comes with this. So it's no problem, of course. So this is it's just quick and dirty, and it's not the most beautiful. It's just also MATLAB has these kinds of functions where we can just quickly plot a mesh in 3D. Maybe in MATLAB it's even easier. I think there's this plot 3 function or so.
Python is better.
Okay.
But thank you.
Okay, these are just my IRB functions. Let's look at this other video, at a video now. So, as I said, this video, I made it, I don't know, 6 years ago or so. It's Jessica's brain. Here it is. Yes, so it starts, and this is Jessica's head surface and also her brain underneath. So here we start with the rotation of her head surface. Underneath we see the scalp anatomy a little bit, and then comes the cortex surface, and then underneath the cortex surface we see Jessica's fiber tracks which connect the brain regions, which we got from tractography. And this is, the tractography was done with the same software package where the viewer belongs to. And so it was a straightforward choice to visualize the tracks with this package, MRtrix or MR view, because it is able to visualize its own files which it generated. So after you perform such a tractography, you can have a look at this tractography at the result of a matrix directly in the native viewer of a matrix. And so the viewer is kind of basic. It doesn't visualize things in the most beautiful way, and it hasn't much features for automation, so it's a lot of hacking. Let's just look at some example data. So here we have Jessica's T1. This is her T1 MRI scan. And the MR view is already able to render this as a 3D volume. This is quite cool actually, that out of the box this software does this. This was also one reason why I took it here for this video. It was a bit easier to work with, but one sees these aliasing artifacts on top, so it's not like the nicest surface representation, one has to say. The other data we have here, so let's just use Jessica's brain. So here we extracted her brain, and now we can add her tracks, her fiber tracks. So this is downsampled basically, but here we can basically look at the fiber tracks overlaid on the brain, and this is a nice first check whether the tractography result is plausible. So if these fibers, no, that's not the optimal view. So if these fibers end up where they should, namely at gray matter, then basically we can be sure that it worked out nicely. So sometimes you see obvious errors where this whole track pattern is somewhere completely off, basically. So this can serve as a quick check whether your tractography result is plausible, basically. And you can visualize and overlay different kinds of data. So for example, here we have a parcellation. Here we have a parcellation where each region has a different color. So each region has a different color, basically. And this could also be good for doing some quick checks, basically, on your tractography output. I think you already saw some sessions about the pipeline. So when the pipeline result, the tractography pipeline, comes to you, you can just quickly look through the pipeline result and make some visual checks whether this is plausible. So in order to construct a video with this software, this was actually quite a pain, because there's no real scripting functionality in the software. So it has the option to perform screen captures, and these were used here. So you have to define some camera position here, some angle where you look at, and so on. And then you basically can also select some kind of rotation, I think, or maybe also not. I think it was possible to select some kind of rotation, but anyway, in order to construct this video, what really one had to do was basically with another software write out commands, MRView commands, and provide these to the command line. So basically for each frame of this video, you need to construct, so a video is nothing else than about 30 pictures per second, right? This is a video. And so basically we need to generate with this software 30 pictures for each second of our video that we want to show. And each of these pictures is basically one call to MRView, and you can basically give MRView all the things that I just did in the GUI as command line options. So for example, here I would load some kind of background image, I would load some kind of tractography result and so on. I would set some view options, the camera position and so on, rotate the brain where I wanted, and then make a screen capture of this image, basically, write it out, and then with another software I put together all these pictures and have a video. So this is quite painful, but yeah, so there's no real software which does all of that for you. Okay, okay, now let's come to the last video, and this is like really the most complicated way to produce such a video, but yeah, and it contains a lot of information. It's very dense, but yeah, it's also the quality is much better. So this was really done with a software that is able to make really nice renderings of brain surfaces. Okay, so I will just start this video here. So this is again Jessica's head surface, and it's much nicer. No artifacts and so on. And here the brain surface is also really nicely rendered. The underlying tracks also. So here we can see this whole information much nicer. And so to produce this video, really we have to script the entire process. So I didn't find a software back when I did make this video who was able to do this. So really it was necessary to hack all this stuff together, basically. So there we saw the abstract brain model and how we arrived at it from tracks and parcellations and so on. And here again we have the brain surface, and on the right side we see resting state network activity. So these are networks that fluctuate all the time. And on the left we see EEG activity also projected on the same surface. And in the middle we see basically how a brain network model predicts the right side from the left side, and how it links basically EEG activity and fMRI activity. And yeah, this is basically an explanatory video that tries to explain the mechanism that links these observations we have, basically. So here the video really tries to explain a mechanism that that we inferred with the brain model, basically. And for that you need, this was basically all scripted together. And I will, in the remainder of this talk, I will basically show you how this was scripted together, more or less. And really each frame of this video basically needs to be constructed and written out, basically, and combined. So this is quite a lot of work and quite complicated, but I will give basically a rough outline of how this could be done, and the general framework then is basically applicable to all kinds of things that you want to represent with a video.
Put the microphone from here to the
No problem.
We can put it like in the you don't have to make a whole other story.
It's no problem. Maybe to avoid that it.
Okay.
Yes, it's no problem.
Okay.
Okay. So this was the video. So I will now first outline these steps roughly with slides here, and then later go into the coding details, basically, and also the data we have here. So step one. Step one is, we saw in the video, resting state networks. Resting state networks are quite an important finding of neuroscience of the last decade, maybe, or even earlier. So when you put people and animals and so on in an fMRI scanner and they just lie there doing nothing, then always the same set of networks pops up, and we have this slow waxing and waning, one could say, of their strength. So they become stronger and then weaker again, and all the time they pulsate on a slow time scale. So maybe in 20 seconds or 30 seconds, such a resting network basically performs one oscillation. And yeah, so resting networks are there are also task networks. So also if you perform a task, there are task networks which show up in dependence of the task you are performing. So for visual tasks, you have strong networks in the occipital areas of your brain, the visual areas. When you have a task that requires a lot of high-level function, you have networks showing up in the frontal regions of the brain, and so on. So these networks can be extracted from fMRI data. This is a common procedure with a method called spatial ICA, which is similar to principal component analysis, which basically finds a decomposition of the data into a set of basis functions, so to say, or components that allow reduced or simplified representation of these data. So when we have our fMRI data, so here we have space. So this is basically a 2D matrix where all the voxels of the fMRI data are put one after the other. So here it's not like in a 3D volume, but we have just our voxel 1, voxel 2, voxel 3, and so on, just one after the other. And here we have the time. And what the spatial ICA does basically is it gives us, the space here, the voxels, we have a large number, right? It's in the order of 100,000 or so, or up to maybe with new fMRI resolutions, even 1 million voxels, right, that belong to a brain. So this is really a lot of information and it's hard to digest. And so the principal idea is to divide this data into subcomponents. So this is how people found out about resting networks. So this huge matrix here is decomposed into a very simple matrix. So here we have only maybe 20 or 30 or 40 components, and how their activity changes over time. And these are the components, right? So to the left here, these are resting networks. These are the components, basically. This is the information that we see here. So when we see here the resting network in a dark color, this means that this component is strongly active at this moment. So also the voxels in this that underlie this yellow patch here will show a strong signal at this moment, or a strong oscillation. And this is this simplified representation of this, basically. And here in these spatial maps, we basically only store at which location is which resting state network. So this is a spatial ICA. Best results I typically got with FSL melodic. So it has a GUI, which is nice to click together a few example cases. The nice thing is melodic will then output the command line in order to reproduce the GUI commands that you just entered. So you can also script this then when you have a lot of subjects. And the best ICAs with really nice, sharp boundaries between resting networks you only get when you perform this ICA on a large number of subjects. So you need to concatenate the information of many subjects. They need to be registered also. And then you get this information, basically. So this is FSL melodic in order to construct resting networks. And what you get after that is basically these nifty files. Has someone told you what a nifty file is? So a nifty file is like the standard brain imaging format for MRI data. So when MRI data comes out of the scanner, it's in DICOM format, but it's a quite impractical format, and quasi-standard in neuroscience, neuroimaging is NIfTI. NIfTI is a simple format where you have just the 3D image cube, so a 3D array of your voxels, basically the amplitude of the voxels over time, so a 4D array, and just a few meta information like the space, the rotation, and so on. And what you get out of the ICA is such NIfTI files where you have for each ICA, for each resting star network, basically the amplitude or the strength of this resting star network for a given voxel. So this first network here is very strong in occipital regions. So no color option. What's that? Anyway, yeah, it's we don't have false colors here, but you see the amplitude is strongest here in the visual cortex. So this is a visual network, and this is basically the information we get here. Okay, next step. Step number 2 is we need to extract the head surface and have it in MNI space. So at the beginning of the video, I did a little trick. So we see Jessica's head surface, her scalp and face and so on. But underneath it's not Jessica's brain, it's not Jessica's cortex. I took here a standard brain, an average brain, because the results of the ICA are all registered to the standard brain. So they are all defined in this standard brain space. And also the standard brain is very smooth. So it's the average over many individual brains. And this is like a common target in order to register or normalize your MRI data to have them in a common space. And yeah, this is what we see here. So we somehow have to bring this together, right? Jessica was at some point lying in the MRI scanner, and we have her head surface in some space, and we have this MNI brain also in some space. So it's somehow rotated, it's somehow positioned and located. So we must bring these two together. So it should look real. It should look like this was Jessica's actual cortex. So in order to do this, one needs first to extract Jessica's brain. And then this can also be done with FSL BET, brain extraction tool. And then when you have this, so here you can see there's quite a difference between individual brains. So this is not a specific feature of Jessica's brain. So every person's individual brain looks much more wrinkled and chaotic, so to say, than this average brain. So this average brain looks much smoother, which is also a reason why it is good for a visual representation. In order to bring these together, we need to register them. So during these pipeline talks, you surely came across some image registration also, where you basically registered diffusion weighted MRI with T1 or T2 and T1 or fMRI and T1 and so on. So all of these registrations have to be performed in order to construct brain models. And here we register Jessica's brain with the MNI brain in order to find the correct transformation rule, right? I couldn't register Jessica's head surface to some standard head surface. A registration algorithm would never deal with that, right? So the best we can get here is take the brain out, register it onto the standard brain, and take the registration rule that we obtained from this process and use this registration rule in order to rotate Jessica's head surface into the same space of like the MNI brain. So when you do registrations, you often have to experiment a lot. It's more like an art than science. So sometimes the registration also works better when the remaining tissue is around and you don't have just the cortex. So you have to experiment with this, but in this case it works best if we extract the brains and map these onto one another. Yes, so when we so in order to get these surfaces, this like the brain surface and the cortex surface, FreeSurfer is a really nice software. So FreeSurfer is like the standard starting point for many neuroimaging investigations because it produces high quality results, high quality surface measures, usually if there's not some defect in the brain. So in patients it often fails, but with a normal healthy brain of an adult it usually works. If the adult is not too old also. And here now comes SurfEyes, which is a nice software to which is the software basically to make these beautiful renderings. So this is really a good piece of software. It is written by someone who wrote a lot of important software packages, and it's high quality. So here you can really do everything, or not everything, but you can do a lot and have a lot of options for automizing this. So we can, for example, that's strange. Actually, something should happen now. Anyway, yeah, it has different renderers, so this is not the only way to visualize surfaces. So it has 1,000 renderers or so, which all give slightly different surface representations. So this is really all the same information underlying, right? But the renderer and the shader in the end make a lot of difference in order to get nice 3D visualizations. And I haven't found another software package so far which is able to visualize a brain like in this meaty way as it just came out of a body. So this is quite nice. We also can play around here with these sliders here. We can also add overlays. Maybe let's start with the different surfaces that we have from Jessica. So we already saw the scalp surface. So this is no. So this is the scalp surface again. But FreeSurfer also constructs other surfaces. For example, we have the outer skull. So this is the outer skull. So this is really just extracted from a T1 MRI image, right? So this is quite a good job for an automatic software. So you really don't need to, mostly need to do anything. Just give the MRI data to FreeSurfer and it will construct these very nice surface representations. The inner skull, yeah, looks a bit not so nice. The pile is basically the cortex, so this is the interface between the pile, the outer skin layer of the cortex. We also have the inner cortex, so this is underneath. This is the surface between the gray matter and the white matter, basically. So here the tracks start and connect brain regions. This is the brain. Here's only the brain. And here is, yeah, the scalp we had already. Yeah, okay. Yeah, to wrap this up, so you get these different kinds of surfaces, so scalp, inner skull, outer skull, cortex surface, and cortex and white matter interface, basically. So you have beautiful.
Can you send me this model?
Sure, of course.
I really want this. Yes, I'm gonna 3D print it because there's nothing wrong with that.
It's your brain, your data. Okay. Yeah, and this is the kind of data you basically get from FreeSurfer. It really produces high quality surface meshes, and together with SurfEyes, you can visualize them in a nice way. We can also just, so other things we can do with the software, we can like open the standard brain again, and then we can add overlays. For example, we just saw these resting state networks that we computed with FSL Melodic, and we can just project them here onto the cortex surface, right? So this is one resting state network. We can also add maybe also a few. No, I can just add single resting state networks at a time. This is some kind of, I don't know, default mode or so. No, it's not a default mode. This is another visual network. So all kinds of networks, you can project them here, give them different color tables, constrain the coloring scheme and so on. So yeah, this is a nice way to visualize resting networks, basically. You can also add tracks here, maybe these. And now we can look inside with this one, yeah, this slider. Let us make the outer mesh transparent, so we can look inside, and we see the tracks in this case. We can also add nodes and edges like we did before. So the software came out some time after I made this first video with MRView. So here we have nodes and edges. We can also play around with the node size, can make them super large, threshold the size based on some information, make the tubes large. So here you can also represent all these kinds of different information in a single 3D object, basically. And the cool thing about the software is that it has a scripting interface. So not the only cool thing, but one of the cool things is that you can really script an entire scene, and even now you cannot script a movie scene, but you can script a 3D scene and basically then vary one rotation parameter in here and script your way to have an animated 3D object, basically. So here you just have commands that do what I just did by clicking. So you would load some kind of mesh, load some overlay, like a resting network, for example, define some camera positions and so on, some view and angles. And then you can also define some rotation values. And then, for example, you can increase. So in order to have a rotation like here, right, you can basically write out such a script. So you can automatically generate such a script using Python or MATLAB or whatever, and write out such a script for every frame of such a video. So here we have a video, and 1 second is like 30 images or so. So you make 30 very, very tiny rotations, basically, in order to get a smooth transition here without much bumps. Okay.
Sorry, one thing.
Yeah.
The format of the file that you give to the surface is nifty, or is it?
The surface is always in this vertices and triangles format. So there we really have only.
So
yeah.
it actually gets like text files.
Yeah. Basically, yeah. It has an own format, MZ3, but internally this format is nothing else than the coordinates basically, and so we have the setup or just the file, the data format is always the same. You have 3D coordinates, x, y, and z, and you have triangles that connect these points, right? And this is really all that you need to specify this. So if you want to specify this, all you need is these two files, the x, y, z coordinates of each point here and the triangles. And between these, or the result of this, whether it looks like this, or like this, or like this, really depends on the renderer and the shader and so on that are used there. So
also the fibers.
No, the fibers are stored in another file format. So the fibers are stored in TCK format. TCK, or it depends on the software that you're using.
Surface.
It depends on the software that you're using to construct the structure connectome. Right, the tracks, exactly, the tracks, yeah. And so, for example, Matrix writes out TCK files, which is basically also just line objects. So a track is a line, and a line consists of points and connections between these points. So the line is basically just represented by a number of points, dots, exactly. Yeah. So this is what is written in the TCK file. And Matrix constructs these files for you, and also it has the ability to write out these files in different formats. So for example, you can use this Matrix function TCK convert and output it in different coordinate systems and different track file formats. So I don't know what kind of formats they are supporting. Yeah, but this is one way. Sometimes you may use one program to get from TCK to some other format and then use another program to again convert this, or you load them with Python or MATLAB and manipulate them yourself. So this is up to you then at this point. But yeah, this is like a common, the matrix format is quite common and can be read by surf-eyes. And the matrix also has a lot of functions to work with these with this track file format. So as you can see here, it has 1,000 functions where you can do all kinds like resampling these tracks, for example. Typically a track file is quite large, but you can also compress it by just including every second or third point or so on, for example, of each track. So this would make it a bit smaller. Yeah. Okay, so this was Jessica's Scarp and MNI. Okay, yeah, we already saw this in surf-eyes. So we construct these rotation images basically, and we also overlay them, for example, with resting networks in order to get this. So it's about this here. So right here we have these resting networks, and they get stronger and weaker all the time. And we do this by loading the brain surface in surf-eyes, constructing resting networks with FSL melodic, and then setting them as an overlay. But this is not like, there's still a bit of hacking, which we'll show in a minute, in order to really get them nicely overlaid. So, for example, surf-eyes can't really show these different amplitudes, right? Whether this is very dark or light yellow here, for example. This I did also manually in MATLAB basically. So we have to, SurfEyes delivers the ingredients, but we again have to put them all together by ourselves. Okay, then one part of the video was a projection of source activity onto the cortex. So in the left panel here, this is EEG source activity. Source activity, what is source activity? So you know EEG activity. EEG activity is you have electrodes around your scalp and they measure electrical activity on your scalp. Now we would like to project the information that we measured outside the brain onto the cortex in order to have some kind of spatial localization, that we somehow know at which point did which kind of activity originate from the surface. So this is an underdetermined problem. We cannot solve it really, but there are heuristics and approximations that allow us to project source, EEG activity onto the cortical surface. And this is called EEG source activity. And this was done with a package called Brainstorm. Brainstorm is a nice MATLAB toolbox for doing source imaging. It has a nice GUI where you can also construct all kinds of visualizations and visual output, and also construct batch pipelines. So you can click together a pipeline for processing for a single subject here, and it automatically generates you scripts that let you run this pipeline over all subjects. So it's really a convenient software. And you typically start by importing an MRI image. That is just a T1 image, and then we also import triangulations of scalp, skull, and cortex. So what Brainstorm does then is it downsamples the cortex to about 16 or 15,000 vertices, and at each vertex position it puts a dipolar moment model basically. So the assumption here is in the cortical surface, the large pyramidal neurons are all perpendicular to the surface, or more or less roughly perpendicular. So if our cortex gyrus is like this, then many neurons will stand perpendicularly on this or under the surface. So we have a crude idea of how the electrical or the electromagnetic field will be. So we construct at each surface location such a model, and basically try to estimate or fit a model that that optimally predicts the EEG activity given this model basically. Then we align the sensors, so we have now a 3D model of a head, including different tissues like scalp, skull, and the cortex beneath us. So we have different shells because these different tissues have different electromagnetic transduction properties. And out of this, basically boundary element method measures are constructed that simulate the transduction of electromagnetic fields through different types of tissues. So what is missing when we have the model of the head basically is a model of the sensors. So here in this case, this is an MEG helmet, not EEG, but an MEG helmet, where we have these sensors here. So we bring these two models together. Then we perform some artifact detection, remove eye blinks, heart rate artifacts, and so on. And then we construct such a source imaging model. So basically we start with a forward model. So as I said, we have these dipolar, the small dipolar moment models where we assume that underlying each surface vertex is a group of pyramidal neurons perpendicular to the surface which have a dipolar electromagnetic field. Using this model, which is an underdetermined system because we have 16,000 locations here, but only a few hundred or a few dozen locations sensors here. So this this is a problem that cannot be fully analytically solved. So we use some heuristics and approximations in order to invert this forward model and arrive at a model that basically takes our EEG activity and projects it onto the cortical surface. And then we can create nice movies like this. So this comes out of Brainstorm. We could also have used this, but we want, of course, have everything in the same coordinate system to make it comparable. So we also had to get this data and somehow project it here onto the cortical surface, which we'll show later how to do this.
Sorry.
Yeah.
Can TVB do the same?
The source imaging?
To go from the source space of the brain to go to the sensor space for EEG?
No. There's no source imaging in TVB. So source imaging is already a very complicated procedure. Yeah, this is basically implemented in Brainstorm in the most nicest way I could imagine. So it would be a lot of work and redundant work to also implement this in TVB. So I would really suggest to use Brainstorm. It's really a nice software.
It's MATLAB. So every time you want to do this, you do the simulation in TVB, then export everything to Brainstorm.
So you need this only for a very confined use case. So you need this only when you want to perform source imaging. So if you have EEG data and want to know what is going on the cortical surface for validation, for example, yes. When you want to project your EEG, your simulated EEG activity on the cortical surface. But I think in TVB, so for example, in TVB, you have this average orientations for your regions also, right? So the input data in TVB consists of...
Yes, I remember there was orientation.
Yeah, so we have average orientations for our brain areas in TVB.
You can just hire them up, right?
And these average orientations are then used, or can also be used, to project the activity to the sensor space. So I think there is some low level, so you can definitely simulate EEG activity in the virtual brain. I'm not sure now how exact this is. So I mean, here you have these average orientations. You would also need sensor locations to fully model this forward stream. I'm not sure right now.
And also the mass conductivity, right?
Yeah, you're right. Yeah. We also need, I'm actually not sure how this is done in the TVB software.
It's okay. Thank you.
But yeah, there are some options to simulate EEG. But what you can do is, after you have, so really a high quality source imaging can be done with Brainstorm. And after you did this source imaging, you basically get your forward model and your inverse model in a very compact representation. So in the end, the forward model and the backward model is just a single matrix, basically. So if you want to go from your 76 brain regions, say, to 60 EEG sensors, then you have a 76 times 60 matrix, and you just multiply your time series. So your time series is something like 76 times 10,000 or so, right? 10,000 for 10 seconds activity at 1 millisecond resolution. And then you just multiply these two matrices and you end up with the time series at your EEG locations. So ideally, maybe not ideally, but a good way is to use Brainstorm to do your source imaging, export these two matrices, and do it outside of TVB. Maybe, I don't think TVB has already facilities to get the same quality of source imaging natively out of TVB.
Thank you.
No. yeah, I just saw that I forgot to yeah, show. So these are basically example images that you would then. So these are for different rotation angles, basically. So here, really, I just rotated the brain with small steps, basically, and this is and had the different resting state networks. So I did this for each individual resting state network. I made a full 360-degree circle, basically, and outputted for each individual resting state network, basically a view of the brain for each rotation angle, and we will later put all of these together then in MATLAB, or this can also be done in Python then or whatever. Yeah, for source activity, there were also some tricks necessary. So for each rotation angle, I outputted the brain completely without anything, then only showing the region centers, then showing region centers and node edges, and so on. So all of this was necessary. Then just showing with source activity, then just showing this kind of activity. So all of this was necessary in order to put this together, then to have this to get this video. We'll see this in a minute. Okay, we have source activity. Here we have. Okay, let's jump over this. We will look at this later when we go into the code. So this is just here we have animated time series plots also. This is also an element of the plot. So at the top you can see that there is a time series changing. So this time series also has something to do with what is shown here. And we also have to construct these time series animations, these videos. This is also basically plots where small elements are added. So we add small points here for each time point and write out basically these plot windows and then later put it all together in order to get this video. Okay, and yeah, let's put it all together now. So first step was just as a short recap. We had extract the resting networks. Then we had to write out rotating brains with resting networks projected on top, just with nodes, just with edges, and so on, because we wanted to arbitrarily combine these different elements. So we overlaid all resting networks for all rotation positions and wrote this out basically, and then later within MATLAB we controlled the opacity or transparency of these resting networks. So here the right, we see that the opacity or transparency of these networks is here we see that it's fluctuating all the time. Sometimes it gets really dark, sometimes it gets very transparent, and we did all of this later than in MATLAB. And this is like in the RGB color model. You really just need to, in order to overlay two different things in a visual space, you basically just add them together and differently weight depending on the opacity, the background, and the overlay image, basically. We also generated node and edge files. So how exactly this was done? So I'm jumping over a lot of small things that need to be done. So this looks also super easy. Actually, it wasn't. There is a lot of small things that need to be done. And I skip over these, so this is just really to give an outline about the major steps. Okay, now let's look at the code finally to wrap this up, how this could be put all together. And as I said, this is really just like the meaty parts of the code. So a lot of things were omitted to make it a bit more digestible. Okay, so here in the first step, very often you need to do some nifty file manipulation, and you don't find a program which does that. So in MATLAB you have, for example, a toolbox which is called nifty toolbox or so, and it has functions to load these nifty images. And after you have this nifty image in MATLAB or in Python, so Python also has toolboxes for using and working with nifty images. So this is a very good skill to have, knowing toolbox in Python or in MATLAB, with which you can load nifty images, look at the file header, the metadata, for example, and really work on the raw imaging data. So this is often necessary because these standalone softwares often don't let you do everything you want. So here we really need to work on the bare metal, basically. Then we generate these rotating brains. So here I basically generate scripts for surf eyes. So here I put together such a script. Here I define some kind of rotation parameters. I say it should write out these generated images, and then I construct here 360 scripts that each call surf eyes 360 times and write out one image. And we do this for all different kinds of different combinations that we just saw. So just an empty brain, then a brain with resonance networks on it, or with individual resonance networks, then with nodes, edges, and so on. We need to write this out to have the full control. We need to write this out for all these different combinations to have the full control over combinations, basically. And this is what we do here then. Now we combine these different images that we wrote out. So after I created these images with Surface, so Surface creates a snapshot, writes out an image, stores it on the hard drive, and now I read this image with MATLAB. Okay, so this is one kind of image. So here for example, I have an empty brain which just shows the surface, and here I have an image which also shows one resting state network, like this, for example. And then I combine these images. So I load all of these 9 resting state networks I have here. Then I look, what is the difference between the empty image and the resting state network here we see. So I select all pixels in this image that belong to this resting state network. And then when I have these pixels selected that belong to each of these 9 different resting state networks, I can put them together. I can produce composite images where I blend them together, basically, using the RGB color model, so red, green, blue, basically. We have these 3 channels, red, green, and blue. And for example, if we want to show, so I have one image and I have another image, and if I want to show both overlaying in a transparent way, so that I have 50% transparency in both images and show them both overlaying, then I would have this parameter I here would be 0.5. And I would then, so I have 0.5 times my background image, and then 0.5 times my image I want to overlay, and I add this, and this is the way how transparent images are produced, basically. This parameter A is then basically, it depends on the amplitude of the resting state network, right? So when we did this, when we extracted these resting state networks here, we got, when we have like 9 resting state networks, we have these 9 components here, and then we have an amplitude, a strength of this component over time. So this fluctuates, each resting state network fluctuates over time, and I basically adjust the visual appearance, the transparency, opacity of the resting state network, depending on its amplitude. And this is what is encoded in this parameter A. And we can combine different resting state networks, different images, and combine different images and overlay them. And after we have this image, then we construct a big empty image. So here I have a big white, empty white image. So the complete frame here from top to bottom. And then I take this new composite image for each frame and put it at a certain location in this. So let's say this full image is like 2100 pixels wide and has a height of 2150 pixels, and then I put it at a certain location in this image. So I merge background and foreground images together, put them at a certain location, and then I use MATLAB, the MATLAB function video writer. So basically, this was the construction of a single frame, right? And then I do this for every frame in this video. So this video is like, I don't know, 2 minutes long. So 2 minutes times 60 seconds per minute times 30 frames per second or so. And this is the number of images you produce. And then you construct such a data structure and store all these images in there. And MATLAB has then a function to basically write these images out as a video. And here we have an AVI video then. So here, this is how a transformation looks like. As I said earlier, when someone lies in the scanner, we have them in some kind of coordinate system. Then we have the MNI brain, for example, in some kind of standard coordinate system. Then another software has its own, so Brainstorm, for example, has an own coordinate system. Then again, FreeSurfer has again a different coordinate system. So each software has their own coordinate system. Each machine has their own coordinate system. So up to now, there wasn't really any consensus. People didn't find a common coordinate system. So we have to rotate them. And even if we would have a common coordinate system, we still would need to register different images onto each other because, for example, when you do one scan, you do an fMRI scan, and later you do a T1 scan, then your subject will have moved in the scanner, will have maybe the brain, the head moved, and so on. So you have to bring these different modalities together, you have to register them. And after you did such a registration, you get a transformation matrix. So in order to rotate a three-dimensional object or to transform a three-dimensional object in a three-dimensional space, you need a 4x4 matrix. So these are 3x3 rotations, and 1x4, so the last column is the last column, the last row are transpositions, basically, where so you rotate with a 3x3 matrix and you transpose somewhere in 3D space the object. And so a rotation and such a transformation operation is nothing else than having a transformation matrix and then multiplying it with your vertices. So here again we have our vertices. As we saw earlier, this is just a text file, or this is just n times 3 coordinates, x, y, and z coordinates. And so this is a matrix, and we multiply this matrix, this vertices matrix, with our transformation matrix, and this gives us then the points in the new space, basically. And yeah, we have to do this over and over again all the time. Sometimes this is already done within some software, so there is software which does all of this automatically for you, but often you have to code it yourself. This is the same. So this was what we did earlier. We transformed, we registered Jessica's brain with an MNI brain and used the transformation rule to transform her scalp and skull to MNI space. Here we generate node and edge files. Here again we have to make some conversions. So this time we use a conversion function from Brainstorm, which transforms also the different involved spaces. And what we do here is basically we export an atlas. As this atlas is very similar to the atlas we just saw. So an atlas is nothing else than a region mapping that associates each of these 16,000 vertices with one number, like 36. So this vertex here belongs to brain region 36. And this is the same what we do here. So Brainstorm also has this atlas, these atlases and these. Oh, do we have a Can I borrow some energy?
Energy? Oh, yeah.
Perfect. Thanks a lot.
No problem.
Okay. Yeah, we do the same basically as we did in this very first example, where we found out which vertex belongs to which region here. So this is yeah, some code in order to do this. And then we need to write out this specific format for surface, but surface has also MATLAB functions which lets us write out this format basically. And here we generate these animated cross-correlation plots. So what these plots show here is the correlation between simulated and empirical resting state network activity. What we do here is basically, and also between fMRI functional connectivity. So we first have to compute the functional connectivities, and then we again correlate these functional connectivities. So yeah, this is just quickly skimming over. So what we do here is we correlate stuff. We produce a time series of sliding window correlations. So basically, we define a sliding window of our simulated time series and a sliding window, a window of our empirical time series. We correlate the two. So this gives us one data point here in this plot. And then we construct these animated time series plots by writing out one image for each time step here, basically. So for each time step of this time series plot, so when this moves, then this time series gets longer and longer and longer. So for each moment in time, we write out one image again, and to make it smooth, we also interpolate between data points here. So just some interpolation operations to pump up the temporal resolution. Then here we just plot it. So this is all just parameters that constrain this plot, the appearance of this plot, basically. So this is nothing important. And here we do basically the same for these small time series plots down there. So here we have also a time series plot. Here we have a time series plot. Here we have one, and here we have one. So for each of these plots, we have to do the very same. We write out an image for each moment in time, basically, and put them later together to a video. And here we do this source imaging. Yeah, we read all kinds of. So after we did, so this is the final step of the source imaging. So this is when we have already this matrix generated, right? So we did a long and tedious source imaging procedure with Brainstorm, and at the end we get a compact representation, a matrix. And we load this matrix here. We also load EEG activity. We also load the high-risk cortex, the low-risk cortex. So here we have to bring together these different cortices. So at the beginning we have in surface we have this very high resolution cortex, but for Brainstorm, for the source imaging, for the source model, we use a low resolution cortex, because otherwise the problem would be even more underdetermined. So here we just use 16,000 surface vertices, while here we use more like 500,000 or so, 500,000, something like this. And so we have to load both of these, and we also have to somehow bring them together to match them. So which vertex of the high res belongs to which vertex of the low res. So this is also a lot of manual work that you have to do here. So this is all pretty hacky, but yeah, when you want to do something advanced, you probably won't find a software which fulfills all your needs. So you have to hack this together, basically. So here is this is a k-nearest neighbor search, basically, for each vertex in the low-res cortex. We look for the closest high-res vertex and construct such a table that associates these two. So here, this is really all. In the end, if we have everything, the source imaging is really just a matrix multiplication. We multiply the imaging kernel with our time series. So this is a time series for n regions. So we have here maybe 76 brain regions, and here we have an No, we have EEG activity here. So this is EEG activity. We have 61 channels and 20 minutes of activity. And here we have this inversion kernel, which takes these 61 channels and projects them onto these 16,000 locations. So we have a time series for each of these 16,000 locations, basically. So this is really how we visualize this here. So each of these locations here. So actually, here we don't have 16,000. Here we have 500,000 locations. So we pump up basically the resolution again by looking for nearest neighbors and bringing it into the space of this high-res cortex. So here, this is really source activity for each vertex location, for each of the 500,000 vertex locations. Here we just make some normalization of the time series. We clip some outliers away and normalize it into some digestible range. And then we write out for each frame, again, we write out one surface, basically, one surface representation. So this is, again, a surface representation which contains the faces, these triangles, basically, then the vertices, and then also, which is what is called here vertex colors, is one number for each of the 500,000 vertices, which specifies the amplitude of EEG source activity at this location, basically. So these are the surfaces that we write out then. And then we again construct scripts for surf-eyes, which loads these surfaces at an overlay, projects them onto the cortex, and writes out an image for each rotation position. Here we associate the parcellation from the low resolution brainstorm cortex with the high resolution cortex. So here we have to transform between the brainstorm coordinate system to the MNI space. Brainstorm has functions, this function csconvert, which does this, so you don't have to worry about finding this transformation matrix yourself. So this is handled internally in Brainstorm, and you just give them your vertex locations. Again, we look for the closest high-res position, high-res vertex for each low-res vertex. And then we basically iterate through our structure connectivity matrix, and we find for each region in our brain network model, brain regions, we associate each vertex of this surface with a brain network model region, basically, and write out again a surface for each moment of this rotation. So this is at the very beginning, right? Here we see the parcellation, basically. So here also for each moment of this rotation, basically for each rotation position, we write out one image with the parcellation here. So we got this parcellation initially from FreeSurfer, then we imported it into Brainstorm in order to do the source imaging. Brainstorm did some transformations with it, downsampled it, and after we had the source imaging, we again export it and bring it again together with the high-res cortex in order to visualize it here. And the final step basically is then, so we had now different, so this were many little videos then, so like this was a video here, this is a video here, this is a video, this is a video. These time series above are videos, these time series are videos, these are videos. So all of these videos have been independently created now. These are all video files on a hard drive. And now we put them all together by creating this big white canvas and putting them at their desired location, blending them, merging them using this RGB overlay. Putting the right rotation at the right position, using the right column maps at the right position, adding arrows, descriptive elements, texts, at some background images. Like here we have a background image that was constructed with some image manipulation program. So for example, you can use Photoshop, GIMP, PowerPoint or so to create images that you can also read then and include in your videos. Yeah, we just add some decorative elements basically, put everything in a video buffer, give this buffer to the video writer and write out one big video. And after we have this video, we may use the software like we may use some software that gets the resolution a bit down, because this is extremely high res. So the video that will come out of such an endeavor will be like many gigabytes large, maybe terabytes even, let's say gigabytes. And then you can, so this is an uncompressed format, right? We really worked on individual pixels here, right? So this is totally uncompressed. So after we did this, we compress it using like MP4 format or something, like using an external software to get it in a digestible size. Okay, I think that's it.